and drug-specific pathway through community structures of a literature network. Okay, welcome.
Thank you. Thank you very much for the organizer for giving me a chance to speak here today. My name is Minh Pham and I'm from Baylor College of Medicine. And today I'm going to talk about a method to discover drug and disease-specific pathways from the biomedical literature. Pathway knowledge is routinely used to explore biological processes in the large omics data, but the current pathway information is very limited. Specifically, it do not cover all of the interactions for the human proteins, and they oftentimes focus more on the general pathway, with very few of them are related to specific diseases and drugs. Manual curation to keep up with the pathway information is the gold standard and preferable, but it cannot keep up with the massive growth of the biomedical literature. And to overcome that, text mining have been used to extract important biological information. And many individual associations of genes, diseases, and chemicals have been extracted extensively, but in order to synthesize pathways, they have to be integrated, and oftentimes through manual curation. And so effort to automate that process have been done, most notably through the BioNLP task in 2013. But most of the tools are very limited to the training data and have difficulty to scale up. Therefore, for our study, we propose a method to combine the manual curations and text mining method. So we know that the community structure in the experimental protein-protein interaction network have extracted meaningful biological processes, and so we thought that maybe we could use that to apply to a literature network. And so our literature network combine the co-occurrences of mesh terms for genes, chemicals, and diseases. And mesh terms stand for medical subject headings, which are curated by biocurators from the PubMed in order to index articles, and therefore they reliably capture the keywords of the articles. And the mesh terms that we use in the network combine the mesh main heading descriptors and the supplementary concept records, which annotate for rare diseases and drugs. Therefore, we captured hopefully the majority of the knowledge from the publications. In order to detect meaningful communities in the literature network, we apply a method called recursive Louvain algorithm, which have performed well on an experimental protein-protein interaction network to extract meaningful biological processes. With that community detection algorithm, we detected groups of highly correlated genes, diseases, and chemicals. Our hypothesis is that the detected literature communities inform on mechanisms of the biological processes and also for the mechanisms for specific diseases and drugs. Our study will allow us to efficiently synthesize pathways from the literature myth associations and also automate hypothesis generation of the disease and drug-specific mechanisms in order to support for disease gene discovery and drug repurposing. So in order to validate the biological relevance of our literature communities, we validate them against curated pathway information. We also explore whether they can capture curated disease pathways and also explore whether diseases in same communities whether they can capture the clinical relations of the diseases in the same communities. Finally, in similar fashion, we also explore whether our communities capture drug-specific pathways. So overall, with the Meteor community and the recursive Louvain algorithm, we detected more than 3,000 communities. All of them have genes and more than 1,000 of them contain diseases, and more than 2,000 contain chemicals. These communities will allow us to explore disease and drug-specific mechanisms. So first, we want to validate whether the literature communities actually are biologically meaningful. To do so, we validate that against curated biological pathway. And here is an example of the biological pathways represented by the ISCB gene sets. We perform enrichment analysis of each of the community against each of the gene set, and we observe that the community p-value enrichment for the ISCB gene sets are more skewed to the significant side than random, suggesting that the communities robustly capture the biological information. We also perform a timestamp experiment to see whether we can predict future pathway information. Specifically, we constructed the communities based on the literature up to year 2005, and then validated the pathways that were added later to the ISCB database after 2005. We indeed observed that there are several communities that were novel early on because there were no pathways added yet, but later when new pathway added, as shown in the blue line here, the communities developed an enrichment for the newly added pathways. An example is that an RNase is a gene set here called RNase MRB complex, which was curated first time in ISCB in 2012, were already majorly annotated by a community constructed in 2005. Overall, these data show that the literature communities predict both current and future biological pathways. Next, we want to explore whether the communities that contain diseases, whether they can capture disease pathway. To do so, we validate them against curated pathways for specific diseases and check whether they have that disease term in those community. We saw that the community especially performed well with OMIM and ClinVar, which are annotated specifically by clinicians, highlighting the clinical importance of the communities in capturing the disease-specific pathways. We also observed that several communities have many diseases in the same communities, and so we explore whether they can capture some clinical relationships of these diseases. We indeed observed that the diseases co-occurring in the same communities have higher comorbidity, suggesting that they share mechanism. Overall, these data show that the communities capture the disease pathways. In similar fashion, we also explore drug-specific pathways. We saw that the communities significantly capture perturbed gene expression profiles for specific drugs, and they capture 50 drugs, while randomly that would be much lower, suggesting that the communities definitely capture non-random the expression profile of the chemicals. We also saw that the chemicals in the same communities also have great shared side effects together, suggesting that they also share some mechanism of actions. And again, overall, this also showed that the communities capture the drug-specific pathways. Because the communities seem to contain very meaningful clinical and biological associations of genes, diseases, and chemicals, we want to explore whether the communities can propose plausible drug and disease mechanisms. Specifically, we want to predict multi-model relationships between genes, diseases, and chemicals. For example, we want to predict the common gene for a pair of diseases in order to understand why the two diseases might have high comorbidity with each other. So intuitively, we would obviously prioritize those genes that are highly linked to both diseases. So that is representing by the number of supporting paper or the edge weight in our network, which is presenting by the term W here, weight. We also propose that the community structure can also capture the associations that were not already mentioned in the network. And that is captured by the number of communities that the entities are detected together, and we prioritize smaller communities, so we adjusted by the sizes. So depending on different setup of our predictions, in general, the community structure method definitely perform well in predicting these multi-relations. And it especially perform well when we combine with the weight. So again, the community structure is useful to detect associations that were not already curated in the, that have not been annotated in the network. We also explore a potential for drug repurposing and by performing a timestamp experiment. In this timestamp experiment, we using the literature up to year 2013, and then we look for disease and chemical pairs that were not found to have any co-occurrence association yet. However, they were detected in many communities together. And indeed, in later year, as we can see here, they were validated. Many of them were validated to be the treatment for their diseases. So these data is highlighting the fact that the literature communities can suggest possible diseases and drug mechanism. In conclusion, the communities, the literature communities not only represented no biological pathways, but they also recapitulate not disease and drug-specific pathways, supporting disease gene discovery and drug repurposing. This is useful for not only text mining scientists in order to narrow down articles in order to explore further the pathway information, but also useful for general scientists who want to explore novel disease and drug pathways. So because of that, we developed a tool, a website with our interface. This is the name of the website. So a general scientist may have a question of like, I have a list of genes and I want to know what diseases or drugs might be related to these genes. So with that, we can enter it into the search text box here, selected them, and we can click on meter clustering or community detection in order to identify the communities. So it's gonna pop up the top hit communities that are significantly enriched for your input. And we can zoom in to see, select whether we want just to view genes or diseases and chemicals. So for this, we see that the input genes are highly related to a number of diseases like ciliopathies and so on. And it's worth noted that this is not the only communities that would be enriched for your input. There are potentially more communities. And to see more, we can click on View More Cluster, and we got a list of other communities that might be enriched for your inputs. We can sort by the enrichment p-value, and we can click on the name in order to get the detail of these communities. So that would give you a list of the diseases and the ID to explore your genes. With that, I'd like to thank you everyone for being here today and for my lab for content support for this project. And I have a poster number H05 this afternoon if anyone wants to stop by. Thank you.
Very interesting work.
Thank you.
So in pathways, as opposed to communities, we usually have a direction, right?
Right.
So one thing is an input to the next. Is there? Do you guys have any interest in trying to figure out orderings or relationships within the communities?
Yeah, so my thought is that the next step is, because we can narrow down the communities, the specific publications that suggesting this community, we can apply the relation extraction tools in order to identify and get more evidence of the direction.
Question?
Yes. So what we've seen in other specific disciplines, such as genomic research, proteomics research, or even in machine learning, is the rapid engage of the community to use your tools if the codes are shared. I've seen great talks today, but I haven't yet heard any information about the system is coded by these languages and you can find these informations at such place.
Yeah.
So I highly appreciate it.
Yeah, so on the website, you can find a download section that have all of the communities that we provided, and we have the network already up there, the clustering method available too, so it's reproducible. Yeah.
Any other questions?
All right, thank you.